Hi, I'm Piyumi. I'm a PhD student at Department of Data Science and AI of Faculty of IT, Monash University. So my project is being supervised by Arun and Peter Starkey and Maria Di Garcia. So the project is about, as you can see, it's about getting physical with proteins and the minimum message length inference of joint distributions of backbone and side chain dihedral angles. So I hope all of us are familiar with the term proteins. Well, proteins are the biomolecules of life because they perform a variety of important functionalities in our bodies. So these proteins are made out of linear chains of amino acids that fold into complex three-dimensional shapes. And the shape of a protein can be expressed as a function of the poses of individual amino acids in these proteins. For an example, you can see here a structure of a protein, this one, and then zooming out an amino acid out of that protein, and you can see the pose there. So the motivation behind this work is to model the distribution of the rotations around the bonds of these rotations around the bonds of the these amino acids, so that these have applications in proteomics. So our method is based on minimum message length, which is a Bayesian information theoretic framework that provides the optimal trade-off between model fit and then the model complexity. So in our case, the model fit is the its ability to explain the underlying distributions, and the model complexity, it contains the number of components and the parameters of those components and the weights associated. So, and the thing is, when deriving these statistical mixture models, we use a unsupervised method, meaning that for our statistical mixture models, we don't know beforehand what is the number of components. So we have a search method that was introduced to search the number of components in the optimal manner, so that we are able to derive the optimal mixture model that is able to explain these distributions of these rotational angles. And here you can see the results of these derived mixture models, and you can see in the blue we are showing the empirical distribution or the observed distribution. For an example, this is for the amino acid asparagine, and for asparagine we can define multiple rotational angles along the bonds of that amino acid, and likewise we can see the distribution, and then in red how well the derived mixture models fit to the these distributions. So in the application wise, these derived distributions have many wide applications in proteomics, like in de novo protein design, and when predicting protein structures, and also when experimentally determining the protein structures as well. So yeah, and also we have made available applications that utilizes these mixture models, so that protein biologists can use them and integrate into their pipelines, and those can be used in their application flows as well. Thank you.